Hi, so this is our project. My name is Nico. I'm currently a senior in BME.
Hi, my name is Joshua Polanco. I am currently a senior in computer science.
Yeah, and we're going to be covering the CNN-based network for gene expression prediction from nucleosome occupancy and DNA methylation. And so just to give background for the problem that we're going to address today, we're addressing the general biological context of development and differentiation of cells and how different aspects that aren't directly related, meaning like directly the gene that's being transcribed that leads to the genetic pathway to confer specific cell fate. So instead of looking at that, we're looking at epigenetic information such as DNA methylation, as well as nucleosome occupancy. And so to begin, DNA methylation, if you look at the bottom right figure, is just the process by which methyltransferase, an enzyme, will methylate cytosine in CpG-rich regions of the promoter, meaning that transcription factors that will want to bind to the promoter region cannot due to steric hindrance, thus diminishing the transcription rate for these genes. And so this is one way that gene regulation is done through an epigenetic pathway. And so this is one part of the information that we would input into our model. And then the next one would be nucleosome occupancy. And so due to the size of DNA, in order to more aptly packed it in cells, it's formed into chromosomes. But the one step back from chromosomes is chromatin fibers, and basically this is just DNA that is wound around nucleosome proteins. And so if the DNA is wound around the nucleosome protein, it creates steric hindrance to regions that are wrapped around this protein, such that gene transcription can't occur. And so the information you receive from this is if a gene is being transcribed, it has to be accessible, meaning it won't be wrapped around the nucleosome. But if it isn't accessible, the gene won't be transcribed at all. And so using this information, we got data from, we got data and we got GNOME-seq data, which is an assay that tests for nucleosome occupancy and also reveals DNA methylation. So on the next slide, this is just a graphic for our model, but the general idea is to take nucleosome occupancy as well as DNA methylation information and input it into a model such that we get a read count. So our ground truth data would be the amount of times a specific gene from the mouse is being transcribed, and use that to inform whoever's analyzing the data what cell determination pathway it's going on. And so in subfigure A, we can see that we have a one hot encoded sequence of the gene, as well as the fifth channel, which is the CpG methylation. And so we chose to concatenate this to the bottom of this array, of this one hot encoded array, because it reveals the methylation rate for the cytosine bases in this gene sequence. And so we didn't include any nucleosome occupancy information because that's implicit in the data that we receive, since the GNOME-seq assay only amplifies regions of the DNA that have been methylated by GPC methyltransferase. And so just to take a step back, what that means is the GPC methyltransferase will go through the genome and methylate GPC residues. And so then the assay will look for these GPC residues, and it is only possible truly when it is accessible and not wrapped around the nucleosome. And so the amplification and the sequence that you actually receive at the end of this assay is just what is accessible, implying that this has the potential to be transcribed. So we implicitly consider that in the model. And then we take this data and we input into a convolutional net and a dense net and output a read count. B just shows more specifics on like the convolution net. Essentially, we're taking two convolutional passes where we do a 1D convolution, batch norm, dropout, and max pooling, as well as ELU activation. We chose ELU activation because it provides the benefits of leaky ReLU and ReLU, allowing it to converge the loss function faster with high accuracies, as shown with some data, and also avoiding any the dead ReLU problem that is sometimes seen. And then that would then flatten it into an array that would go into a dense net, upon which the weights and the process of going through a fully connected dense net would output a single value that represents the read count, predicted read count, which then we would then use mean squared error loss compared with the ground truth and update our model.
Great. And so then with that, we came to pretty good results for our process itself, relatively speaking. So with that, we were to get our model for great running, we would have to create a data pipeline. Given that our data were coming out very raw and not curated. So with that, we had to access the information specifically through special tools given by these databases, one of them being SRA. And with SRA, we were able to get a list of sample of interest, and with that, download such data and format it and have the special format for gene counts and as well as locations. So with that, with the data pipeline, we were able to retrieve the data, the large amount of data, and put it into our special VM, given the size. But then one of the challenges was aligning such data to the specific cells that we were using or the RNA sequences per sample used, as well as their GNOME-seq and RNA-seq to better align the data. So with our data formatted, we then also outlined our model using inspiration from past work in the given field, as well as from our homework. And so then with that, we believe that this was a great starting point, and as such, a baseline in which we can then specifically choose specific parameters and formats to improve on. And then one of the things that we were able to then accomplish after understanding our model was understanding how can we format our data specifically, including the methylation rates that we were given from supplemental data of our GNOME-seq. And so then with all those things together, we delved right deep, and then diving deep, we had some challenges. So on the next one, these are a couple of the challenges that we faced, as well as what we believe theoretically could have been our improvements or where we could have improved from the start. With that being said, the biggest challenge of all was the data retrieval, given how large the data was. However, we understand now that we could have then selected a specific subset of that data to use rather than the immense amount of data that was at our disposal. And with that, the challenges of data remodeling, understanding how FASTQ data formats, the regular formats that are the most common formats used for sequencing, how to take that and put it into a better and more approachable format for the data and for our code to access. Given the sparsity of our data, we could have done a stronger analysis on that to then really decide exactly which sets or subsets of the data can allow us to train properly for our given task. So some of the other extra improvements that we could have done is actually seeing if alignment of these GNOME-seq data to genes could be of improvement. One, for the sparsity of the data, but then as well as aligning more information for the model to pick up. So then, as well as that, we want to improve on variating the model parameters in such a way to improve the model. One of our initial proposals would be to take an initial baseline of the model, seeing how the kernels are picking up information, and then improving on how can we make it extract it in greater depth, or where is it falling off exactly. And the third thing would be seeing if we can explore a little bit more on some baseline sequential based models like RNN, as we've seen in homework 3, to really see if positional information is truly important in a sense for GNOME-seq, given the implicit information that comes with it and the way that our data is formatted. And so then as such, even though we faced these great challenges that hindered us in some moments, we went through a great series of problems that really challenged us to think thoroughly and understand the context rather than just looking at the code. And I think that is one of the biggest experiences and things that we can know about going into the future, is really understanding the context and then exactly extracting as much information from that context.
Yeah, and so thank you for listening to our presentation, and we'd like to apologize for the performance of our group, but we hope that this explanation was sufficient. Thank you very much, and have a great night.